So now that we are done with the align and the quality control and quality trimming process, alignment is the next step. So we are going to align against the reference genome, which we need a FNA file for, and then we need to have a reference annotation file, which is GFF3 file or a GTF file. So after that, we are going to combine this all, and then we're going to call some variants in it. So moving on. So alignment against the reference genome. It's the third step in our detailed variant calling pipeline. So there are different type of tools that are available for the different kind of alignment process. For example, these are called aligners. For example, we need the Bowtie tool. It's a ultra fast memory efficient short read aligner for Illumina data. So we can use the Bowtie. Then we can use the Mr. Fast. It is that allow the multiple location for CNV detection. So what is CNV? I told you that CNV was the copy number variations. So if we need to map the copy number variations, then we can use the Mr. Fast tool. If we need the Corona Light, or Corona Light is a tool or shrimp tool that we can use for the solid platform, which is also a sequencing platform. So if we are using the data that we don't know where we took that data from, or our client doesn't tell us what kind of data we had and something like that, and now that we need to analyze that data. So what we need? So first we need is the tool that can do all that analysis on one, like BWA tool. It's a Barrow's Wheeler aligner for Illumina, Solid and 404. All the multi-platform data that we can get, it can analyze all that. So which tool we are going to choose and why we are going to choose that tool, it's a different kind of story. Let me tell you. There are many aligners, they vary a lot in performance. For example, some are speedy, some are accurate, some are memory, not that memory efficient, some are memory efficient. So which factors to consider when choosing a tool? So first one is the application, why you are using this data. Or okay, so why are you using this data? Which platform you are using, and what kind of read length you require. So there will be a constant trade-off between the speed and sensitivity. For example, there will Bowtie and Mac tool. If you use the Mac tool, there will be a trade-off between your speed and sensitivity. Like Mac is speedy tool, Bowtie is not that speedy, but Mac is that sensitive, it's speedy, and Bowtie is that sensitive but not speedy. So there is a lot of trade-off between your speed and your accuracy. If you use accuracy, you have to go with the less speed. And if your data is not that accurate and you doesn't know that data, so you have to go with all that. So guarantee is that higher accuracy will take a lot of time, and higher accuracy will take a lot of time to process. So for each read, we are interested in the position and the position of mismatches and the variants that we call. So we need a top. So we need a fast algorithm. We need the tool like Bowtie, BWA, and Tophead. So what we are using? So we will be using Bowtie and BWA. Bowtie is a ultra fast memory efficient tool for the short reads aligning and to the long reference sequence. For example, it works with the 1000 character as well. So it is suitable for the long genome, for example, for the plant's genome, but BWA is significant for using all kind of data. So it's keep the indexing algorithm to its memory footprint. For example, it keeps the memory footprint small. It has indexed the human genome around 3.2 gigabytes, which is nearly about 40 gigabytes of data. So how to index the genome, how to use command line for indexing the genome. First, we need to install that tool. We need to install BWA, we need to install Bowtie2. So using BWA, we will be using these two commands to get our data and create a alignment file. But first we need to know how to download a data, how to download Genome. I don't know if you are familiar with it, but let me just tell you NCBI Genome, about NCBI Genome. If you are working on bioinformatics, you are familiar with the NGS data, you should know that how to retrieve a genome. For example, we are working with, for example, human. Let's just open the human genome. So how to retrieve the human genome? So for the first file that we need, the FNA file. So we're going to download the FASTA format for our genome. So we're going to just download the FASTA format of our genome. After that, we need the genome annotation. Then we're going to download the GFF, GTF or GFF3, with whichever kind of file that you need, you can download that. After getting your files downloaded, just paste it to your repository like I did for the genome.fa. If I can open it in my terminal to show you that it contains the sequences, cat genome.fa, it contains all the sequences. So I'm gonna terminate it as well and then clear the screen for you guys, and then we're going to move along. So then how we can use the pipeline to index? So we need the tool BWA. How to install the BWA tool? So simple command as sudo apt install BWA, something like that. So here we are going to see if BWA is installed. Yes, BWA is installed. We are going to use that command, paste it here. Here you can see I have set up the tool index option and then given the name of genome to our indexed genome. So what basically indexing is, what is indexing that we are doing? If you are not familiar with indexing, when this is now it's creating index. So let me just explain you the indexing concept. So sorry for that. So yeah. So what is indexing? Indexing is widely used in bioinformatics workflow for the performance improvement. For example, for large data set, for large files, we need to have many records. For to improve that ability of data, we need to rapidly access some random locations of the file. So we need to have to keep an index of that so we can divide that section. So indexing is basically division in bioinformatics. For example, if we have the alignments against the reference genome and we want to visualize those alignments for a single gene, for example, in CHR17 chromosome, in chromosome 17, we need to visualize that genome by using the IGV. I'm also going to move you to the IGV viewer as well. So we don't have to view and scan through the entire file. So indexing allows us to jump to a correct place in the file and pull out the information we need without reading much of that file. So this is the indexing concept. Like in example, for example, in the book that we start to read, there is index in the first 2 to 3 pages, there are index pages. So we can keep an eye where the main thing is, where is the main chapter, where is the second main chapter, something like that. Now you got the idea of indexing. So after indexing, yep, indexing is done. There will be 4 to 5 files created with the name of genome. Genome, okay. So we have that genome.annotation, genome.bwd. If we can read that file, cat genome.bwd. Yep. It's a machine readable file. We can't read that file, so it has indexed according to the machine's requirement. So after building the index, we need to align that reads to our genome. So we are going to align that reads to our genome. For that we are going to use the MEM option. After that we are going to align that genes and create a SAM file. So it has created a SAM file. Let's just read that SAM file. Here you can see the SAM file is created. This is the SAM file format. It has given us the options of alignment and the scores and something like that, which we can read. It's a human readable format, SAM format. So now that we know the indexing and the indexing concept and the aligning concept, now that we have created a SAM file, how to create a BAM file is another option. So in the next lecture, we are going to see how to use the SAM tools to create BAM files, how to deal with SAM files and how to deal with BAM files as well. So that was it for this lecture. I hope you understood how to index a genome. After indexing, what indexing really is in bioinformatics and how to align the reads of our genome to specific SRA file that we retrieve, FASTA file that we got from our company and our sequencing agency.